Hello everyone, my name is Otman Bouchareb. I am the CTO and director of Inscoper, and I would like to thank you for your participation to our webinar today. So for this webinar, I planned to show you some features of the Inscoper solution, and for that purpose, I will run two experiments within the Inscoper user interface. So the first experiment will be dedicated to FRAP imaging, and for that I will use a sample with U2OS cells. So this is a human cancer cells, and they are inside 8-chamber glass holder, and those cells are transfected with GFP and labeled with oxt at 37 degrees. And the second experiment, I will run tiling acquisition on multi-well plates and on a slide too. With the slide, I will use a Brightfield channel, and for the multi-well plates, inside I have HeLa cells fixed and labeled on the nucleus with oxt protein. So let's start. First, I will present you the microscope we have here in our office. So the microscope I will use for the experiment, and then I will share with you the user interface of Inscoper. So the microscope here is based on a Nikon stand, a motorized Nikon stand with XY stage. We have two SCMOS camera. The first one from PCO, it's a Panda camera, and the other one on the other side, it's an Hamamatsu camera, Orca Flash 4. On the back side of the stand, microscope stand, we have a photomanipulation head here for FRAP experiments. And on the back, we have a 405 laser for photo bleaching, and we have also a multi-LED source from LumenCore. So now I will start the user interface. Okay. So the first window gives us the choice to start a new project or to open an existing one. The user interface also has a feature, an autosave feature, which save all the settings, all the user settings every 30 seconds if we forgot to save our project. So I will start a new project. So the user interface suggests a user protocol or a user journey in order to help the user to run his acquisition. So the user journey is based on 3 steps. The first step is configuration. So on the left side you have the camera view with basic settings like the exposure time and the binning, and we have also access to all the advanced settings of each camera with a filter field. So here we can also use this field to look at a specific parameter for example, of the Panda camera, and we can use exactly the same thing for the Hamamatsu camera. So here also we can create presets of the different settings and save that as a list, and then when we click on that list, the user on an element of that list, the user interface will set all the value of all the parameters of that preset. The user interface also can deal with up to 4 cameras. So here you have the list of all the cameras available. Then on the right side you have access to all the motorized parts of the system. So here you can see the objective available on the microscope stand. There is also the joysticks for the different stage, And here, as you can see, we are on user mode, so we have a limited access to the available devices. And we can also go to the expert mode, and here we have full access to all the available devices. So here you can see the motorized part of the microscope stand, the available shutters, and also different excitation settings. And here you can see the available settings for the multi-LED source from LumenCore also. The purpose of this step is to create channels, okay? To create channels we will use during the acquisition step. Inside the configuration tab also we have different calibration process. So for that microscope we have calibration process for FRAP and for timing. So now I will start with the first experiment, which is a FRAP experiment on live U2OS live cells. And for that, in order to be able to use the photomanipulation head, we need to do a first calibration. Inside the photomanipulation head, there is two scanners, which allow us to target a specific position on the camera field. We need to do calibration because the field of the scanner is much bigger than the field of the camera. So the idea is to make a correspondence between the scanner field and the camera field. And for that we use a fluorescence slide on the microscope. So first we start the live, then it's already calibrated, but I will show you how we run a new calibration. I will create a new calibration, then I select a channel for that purpose, and now I need to focus my slide. Okay, so this is the laser point. Okay, so the idea for this calibration, there is two steps. One manual step. So for this step we need to move the laser point to the camera point. So we need to do this 4 times. So we move the first point, then we select the second one, the third one, and then the last one. Then we select the channel for the automatic calibration, and during this automatic calibration the user interface generates 25 points. Okay, and at the end of this automatic calibration, so the photomanipulation head is calibrated, and we can start to show or to target specific zone of the camera field. So here, for example, I will display a line and a rectangle, and so here you can see how it looks like, and we have different settings to modulate the laser power, or to change the density of the points inside specific lines of the selected forms. So now the photomanipulation head is calibrated. I will put my live cells on the microscope in order to show you how we run a FRAP experiment on live cells. So So now I put the sample on the stage, and in order to focus I will select the Brightfield channel to find my cells. So here they are. I will switch to Ox channel to do a better focus. And as I said in the beginning, the cells are labeled with GFP. So in order to show you how it looks like in live, I will try to find an interesting cell. So for example, that one. So I will do a crop in order to have only this cell on the field of view. Okay, then I will focus a little bit. Okay, so what we should see during the acquisition is something like that. So I will draw a line across the cell, and then I will send the laser within that line, and then as you can see the dynamic inside the cell, in fact. So here you can see that on the line I photo-bleach, there is a dynamic from the other zone of the cell to that line, and other parts of the cell lose their fluorescence. So now I will try to do the same thing but during the acquisition. So I will remove the crop. So here I have the entire field of the camera, and I will try to find another cell. So here they are. Okay. So I will crop the camera again to only have those cells on the field, and then I will focus a little bit. Okay. So then I show you the second step of the user journey. So the second step is the acquisition tab. The acquisition tab, within the acquisition tab, we have an approach of multi-sequence. Okay, so here, and we can add as many sequences as we want. Inside each sequence, there is dimensions. So the first one is time for time-lapse experiment. We have the position dimension for X and Y experiment. We have the Z-stack dimension. We have the multi-channel dimension. We have also an autofocus option. And we have the photo manipulation dimension too, we will use during this first experiment. And we have also other dimensions for other acquisition or for different acquisition, like the film imaging, for example. We have a dimension for film. So for the FRAP experiment, I will define the 3 steps. I will do a first time lapse, then I will set up a bleaching sequence, and then a third step is a second time lapse experiment to see the progress of the dynamic inside the cell. So the first step. So it's the first time lapse. So here I will take 50 images. Okay, and I will define the channel. So the channel, I will select the GFP channel. Okay, and with 100 milliseconds as exposure time. So then I will create a second one and the third one. So here I only copy the first sequence, then I will change the name. So the second step, I will not use the time and the multi-channel dimension, but the photo manipulation channel. So here I will add a photo manipulation. I will use a photo manipulation dimension, and for that I will use the photo manipulation channel. Then I need to draw the region of interest. So I will draw on the 4 cells. So here I will select region of interest 1, 2, 3, and 4. Then I will define a pulse time, which is the time we will target every point in microsecond. So here, let's say 1 microsecond, and the pulse iteration, let's say it's the number of time we will target the region of interest. Okay, and then I will set up the third step. So here I will take, let's say, 300 image with the GFP channel. So at that time I need to select the processing I want to apply to the image. So for that experiment I will select raw data, so the user interface will display raw data coming from the camera. And here we need to define if we want to save image on RAM or on the disk. So I will select the disk to keep the image. And we have also the possibility to generate 1 TIFF per image, or one file for all the generated images. And then here, when I start the acquisition, all the acquisition will be loaded inside the Inscoper box, and this is the Inscoper box which will run independently the acquisition, and the user interface will retrieve the image, will retrieve and display the image from the camera. So let's start. So here you can see the first time lapse. Okay, and then you see the bleaching time. Okay, and then the evolution of the intensity inside the bleaching area on each cell. So here, as you can see, on the first one, which is this one, there is a small evolution. We have a bigger evolution on the second one here, as you can see. So let's analyze this. So the third step on the user interface is the visualization tab. So here we have access to all the acquired images for the different sequence. Okay, we have also filters here to select a specific part of the acquired sequence, and we can also export the sequence in video or in stack if we select RAM on the second step. We have also the possibility to show graphs. So here we show the evolution of the intensity for each line here, and for example, so here if I remove all the region of interest to keep only one, so for example, sorry, I will draw a line. So here, as you can see, that we began with this evolution of the intensity for the first time points, and then you can see the dynamics, so an increase during time of the intensity on the photo bleach zone. So we can also play the video with this button, so you can see here. So on this side we began to see the dynamic here. So I will draw a second line here for you to be able to see the difference with other parts of the cell. So this is the first experiment, and then now I will show you other features of the user interface. So for that I will run a second experiment for dedicated to tiling imaging. So for that I will change the sample, and then I will show you how we set up a tile acquisition inside the user interface. I will also, for that purpose, change the objective, so I will use the 10x for this second experiment. So the first sequence I will show you, I will use a rapeseed flower as a sample. I have a sample with a rapeseed flower, and for that I will use the Brightfield channel. So now I need to focus the sample. Okay, so the sample is right there. So in order to set up a tiling experiment, so I will disable the time and the multi-channel, and I will switch to tiling dimension. So I will enable this dimension, and then I will add a new tile. So here. Okay, and so inside this tile we need to define the two corners of the tile. So I move the stage with the joystick. So let's say this is the first corner, and then I will look. So I will define the second corner. Like this. Here we can also define an overlap, so let's say 10%, and we can choose the read mode or the snake mode for the stage. I confirm. So I will disable the first and the second sequence here. And what is interesting here is that we have shading correction algorithm. So I can show you how we use that algorithm. So in order to do this, we need to get or to take an image as a reference for shading correction. So for that, I will try to move to a zone with no sample. I will change the move out of the focus. So let's take this image. Okay. And then here as a data processing, I select in fact this choice. So we will have a tile with a shading correction and another tile with no shading correction in order to be able to compare both tiles. So let's start this acquisition. So here you can see the tile. So this first one is and this is the second one. So as you can see, the first one with the shading correction algorithm, we get this result. Okay. And the second one with no shading correction, we get this result. So it's very useful in that case to use the shading correction algorithm. What we have also here is a stitching algorithm too. So I can apply it on the first tile with shading correction, in order to be able to compare the image before and after the stitching algorithm. So here, So you can see the difference between the two images. So we can see here that the tile is well stitched, and we need in fact to increase the, for example here, the intensity of the Brightfield channel in order to be able to get this part too. Now I show you an example of tiling experiment on a slide, and now I will show you how to make tiling experiments on multi-well plates. So for that I will place a multi-well plate on the microscope, and I will move back again to the 60x objective for that experiment. So now I placed my multi-well plates on the stage, so in order to run a tiling experiment on that plate, first we need to calibrate the multi-well plate. So this multi-well plate is already calibrated, and in order to calibrate a new one, all you have to do is to get the information from the plate manufacturer, the size, so the number of the well on X and Y, and the gap between two wells, for example, and the diameter of each well. Then we save, and here when we do this, in fact here we have an option for multi-well plate. So here we have a view of the multi-well plate, and we can select any well we want to use, in fact. And in order to completely finalize the calibration process, we run a find center process on any well of the multi-well plate, and the user interface then will calculate the center for that well and for all the wells of this plate. So now I will move to the well B10, because I know that there is some cells here, and I will start the live with the Brightfield channel in order to focus the sample. So here they are, my cells. So the cells are labeled with the host protein. So here you can see the cells, and here, in order to be able to start or to run a tile experiment on that well, it's the same thing. So I will define an overlap of 10% and choose the snake mode. So when I click clear and add, the user interface generates the tile. So here you can see the number of the positions here. I will confirm. So then I will choose the raw data and tile. So here the user interface will display the during the acquisition the tile. So let's start this experiment. So here you can see the progress of the tile. So during time, here on the right you can see the progress of the acquisition. So you have access to the frame rate, to the remaining time, and the total duration. And what is interesting is that the user interface in fact generates very useful data during acquisition. So I will show you two examples. So the first one, we call it the performance file. So here, so inside that file we found all the called function during the acquisition with the corresponding execution time in microseconds. So for each image we can calculate and measure the time needed for to acquire each image. And for example, for that one, it takes for 332 milliseconds. So we have 100 milliseconds as exposure time, and the rest is mainly the time for the stage to move from a position to another position. So it's a very powerful file to measure the latency of a microscope, in fact. So here we know that it's fast, and we know that we can also optimize more the acquisition speed on our microscope, for example by using a piezo stage, an XY piezo stage. So we have that for all the function of all the device available on the system. So this is the first example. Then the second one I want to show you is the acquisition file. So here, in fact, we summarize with text, with words, the generated acquisition and the executed acquisition by the Inscoper box. So here you can see all the steps, in fact, for a specific acquisition. What is interesting too to show you is that the user interface here, because I choose the hard drive, so here the user interface, in fact, save the image in TIFF format, and with the saved image you have also access to the metadata file at the end of the acquisition. So this acquisition is in progress, but I will show you that on another one. So for example, this one. So here you have the metadata file with all the settings for all the device. So this is the generated tile. So here what is interesting is we can generate another tile from that tile. In fact, we can also see a specific part of this tile on live, for example. So here you can see we can also apply LUT, a specific LUT for the image. And then I want to show you an example. So I will generate another tile from that one. So we can define here read mode or snake mode. So here I have a second tile, and I will associate that with a Z-stack dimension. So here I will go inside, and then I will start the line, and I can use the focus map to move to a specific part of the sample. And even, in fact, the focus map is very useful if you have a 3D sample. So you can add focus points to your focus map, and the user interface will generate an linear interpolation for all the XY position. So a linear interpolation of the focus for all the XY position. So here I will confirm. Then for the Z-stack, we can define a minimal plan and a maximal plan, or define a center plan. So let's define a center plan. Then we need to define stack size. So I will say 8 plan with a step, so 1 micrometer between 2 successive plans. So then I will start again the user interface. So here you can see that for each X, Y position of the tile, the user interface take here, you can see the progress 8 plan, and we can compare different style after this acquisition. We have also an autofocus routine, so we can also enable this option if you want, and use it instead of the Z-stack. So here on the right, you can see the progress of the acquisition, like before. And now here on the visualization tab, you can see the 8 tiles corresponding to the 8 Z plans, in fact. So that's it. I hope that you appreciate our webinar and the experiments I run for you. Don't hesitate to ask your question now, and we'll be happy to answer them. And we are also open to your suggestion. And I hope that you appreciate the Inscoper environment. Thank you.